microbial origin occurs by ingesting a food containing a preformed toxin. Some general characteristics of food poisoning are the toxin is produced by a pathogen while growing in a food. Next is a toxin can be heat, labile, or heat stable. Ingestion of a food containing active toxin, not viable microbial cells, is necessary for poisoning, except for infant botulism, in which viable spores need to be ingested. Other than that, symptoms generally occur quickly as early as 30 minutes after ingestion. Symptoms differ with type of toxin. Enterotoxins produce gastric symptoms and neurotoxins produce neurological symptoms. Last but not least is febrile symptom is not present. Food poisoning will lead to foodborne disease such as human gastrointestinal disorder. The causes of foodborne gastrointestinal disorder can be broadly divided into three groups. The first one is from consumption of food and water containing viable pathogenic microorganisms or their preformed toxins. Next is from ingestion of pathogenic algae, parasites, and their preformed toxins through food. The third one is for reasons other than viable pathogens or their toxins. Some of the factors included is ingestion of toxins naturally present in many food. These include certain mushrooms, some fruits and vegetables, and some seafood. Toxins form in some food. Examples are some biological amines that form in some fish, cheeses, and fermented meat products. Other than that, the presence of toxins chemicals in contaminated food and water, such as heavy metals and some pesticides. Allergy to some normal components of a food. Some individuals are allergic to gluten in cereals and develop this digestive disorder following consumption of food containing a gluten.
Foodborne infections. Foodborne infections occurs from the consumption of food and water that contaminated with pathogenic bacteria and virus. There are many pathogens included in this group, but I will share only three of them. Firstly is Salmonella. Salmonella are natural inhabitants of the gastrointestinal tract of domesticated and wild animals, birds, pets, and insects. They cause salmonellosis and then remain in a carrier condition. Humans also can be carriers of infection and shed the pathogens in food by feces. Upon ingestion of Salmonella bacteria, the pathogens attack small intestine mucosa, then mutate in the epithelial cells and produce a toxin that induces an inflammatory reaction and fluid deposition in the intestine. The symptoms of Salmonella infection are abdominal cramps, diarrhea, nausea, vomiting, chills, fever, and prostration, and the disease caused by it is typhoid and paratyphoid fever. However, Salmonella infection can be controlled by proper cooking of foods, such as in pasteurization that use 71.7 Celsius for 50 seconds minimally, and prompt cooling that use 3 to 4 Celsius or freezing if the food are not used in 2 hours. Next is least monocytogenes. Least monocytogenes is isolated from many environment samples such as soil, sewage, water, and dead vegetation. It is also isolated from the intestinal contents of domesticated animals, birds, and humans. The foods like uncooked meat, milk, egg, seafoods, fish, leafy vegetables, and tubers are containing least monocytogenes. The pathogens in least monocytogenes attack different body tissues and mutate inside the body cells, then produce the toxin. This toxin causes the death of the cells. The symptoms of least monocytogenes for normal health people sometimes did or did not produce symptoms. Can be detected in 1 to 7 days and include mild flu-like symptoms with slight fever, abdominal cramps, and diarrhea. Then the symptoms will be severe in a few days by shedding through the feces. But different for sensitive groups include pregnant women, unborn fetus, infants, elderly people with reduced immunity, and people taking special medications. The initial symptoms are enteric with nausea, vomiting, abdominal cramps, and diarrhea along with fever and headache. Then the pathogens are spread widely through the bloodstream and attack tissues in different vital organs, including the central nervous system. Least monocytogens are impossible to get rid of it, but can prevent it with washing raw vegetables before eating, keeping uncooked meats separate from vegetables, cooked foods, and ready-to-eat foods. Not consume raw milk or foods made with raw milk, and washing hands and kitchen tools after handling uncooked foods. The last one is shigellosis. Shigellosis only affected in human and some primates, where it is transmitted directly through fecal oral route or indirectly through fecal contaminated food and water. Humans carry the organism in the intestine and shed it through feces without showing any symptoms. Some individuals may remain a carrier for months. Due to ingestion of contaminated foods, the symptoms may occur in 1 to 3 days, but for mild infection, the symptoms may last 5 to 6 days. An infected person will face symptoms like abdominal pain, diarrhea mixed with blood, mucus and pus, fever, chills, and headache. Foodborne shigellosis can be prevented by individuals by practicing good hygiene and forbid them to handle foods, especially the carriers. The food handlers must use of rigid sanitary standards to prevent cross-contamination of ready-to-eat food, use properly chlorinated water to wash vegetables to be used, and refrigeration of foods to reduce foodborne shigellosis. Thank you, that's all from me.
Disease symptoms of several pathogens with foodborne and waterborne gastroenteritis are different from normal food poisoning or foodborne infection. Okay, in this topic, which is foodborne toxic infections, there are 4 causes of gastroenteritis. Firstly, it is because some foods that were prepared in advance by heating and keep them warm for several hours, for example in the restaurants and cafes. Other than that, the cells need some amino acids for growth, and that is why they can grow very effectively in many protein foods. More than that, the spores and vegetative cells are found in soil, dust, intestinal contents of animals and humans. There were many foods that contaminate with these sources. Furthermore, enterotoxin associated with foodborne disease is a heat-resistant protein. It's also produced in the digestive tract. The main symptoms are diarrhea and abdominal pain. There were also nausea, vomiting, and fever, but less common. Raw meat from animals are commonly contaminated with spores and cells, while vegetables get them from soil and dust. Plus, there were most commonly factors, which are improper holding temperature, inadequate cooking, and contaminated equipment. Next, spores and cells of bacteria can get in soil and dust. The strains produce 2 types of enterotoxins, which are emetic and enteric. Symptoms include abdominal pain, profuse watery diarrhea, and perhaps nausea. When the toxic is stable, it forms in cells and heat the food before produces symptoms, which are nausea, vomiting, abdominal pain, and diarrhea. There are many foods that contain spores and cells of bacteria. However, in emetic form, it involves starchy foods such as rice. It is because after the rice being heated and then keep it at room temperature, the spores and cells still leave before the rice is served. Besides, cholera cause of wheat cholera is a non-contagious disease but can lead to death. But improper heating at low temperature for a short time may not be able to kill all the cells on the food. Plus, alkaline foods make a rapid growth. Cholera is a human disease that causes of weak choleric cells through contaminated
food and water. The symptoms include sudden onset of profuse watery diarrhea and vomiting that cause of dehydration. Other symptoms in worst cases are painful muscle cramps and clouded mental status. Moreover, the food that is from natural reservoirs of the causative bacteria can be contaminated and spread the cholera. Lastly, it produces diarrheal disease when ingested contaminated food and water. It has similar symptoms like in cholera. These strains can establish in humans small intestine without producing symptoms. The symptoms include diarrhea within 2 days. However, in worst cases will be dehydration, frustration, and shock with diarrhea. There are many types of foods which are fish, dairy products, meat products, vegetables that have been associated with gastroenteritis E. coli. Direct or indirect contaminated food and water along with improper storage, temperature, and inadequate heat treatment.
There are several other bacterial species that are suspected of having the potential to cause food poisoning. Normally, they are not pathogenic to humans, but strains in these species have been known to produce toxins. Thus, consumption of foods contaminated with these bacterial species and strains may cause illness, especially under certain circumstances, such as if they are consumed in extremely high numbers, or the individuals are either very young or not in normal physical conditions. These bacterial species and strains are considered opportunistic pathogens. First is Aeromonas hydrophila. These species strains are found in both saltwater and freshwater environments and are pathogenic to fish. Their growth temperature ranges between 3 and 42 Celsius. They are facultative anaerobics but grow better in an aerobic environment. Because of the nature of its normal habitat, Hydrophila is found in many foods, especially foods of animal origin. Hydrophila strains have been suspected to cause gastroenteritis in humans, especially when consumed in large numbers and when individuals have impaired health. Next is Plesiomonas shigelloides. This bacterial species is a gram-negative rod, facultative anaerobic, motile, and non-sporulating. The organism is isolated from the intestinal contents of humans and warm and cold-blooded animals. It is found in fresh and brackish water and in fish and oyster harvested from water. Most strains grow between 8 and 45 Celsius. This bacterial species also can be present in raw foods of animal, bird, and plant origin. They can grow rapidly in most foods under optimum growth conditions, and they are not expected to grow in foods held in refrigeration. Shigelloid strains were implicated in many human gastroenteritis outbreaks associated with contaminated drinking water. Next is known as Escherichia coliforms. These bacterial species are normally present in raw food material as well as in some pasteurized foods because of phosphate contamination. They can grow in many foods if the growth parameters are not limiting. Lastly is toxigenic psychotrophic bacillus. Cells and spores of many bacillus species can be present in non-heat treated foods. Also, most heat treated foods can contain their spores. In refrigerated foods, spores of psychotrophic bacillus can germinate and outgrow, and the vegetative cells can multiply. Some mesophilic bacillus species, such as several Licheniformis strains are toxigenic and are suspected to have the potential to cause food poisoning, especially in foods stored or temperature above at less than 10 Celsius.
First, change in lifestyle and food habits. In this category, several factors can be included that have contributed to the emergence of new foodborne pathogens. One of the most important factors is the increase in traveling, especially international traveling. A person arriving at a foreign country or returning from it can bring a new foodborne pathogen into a country where it was not recognized. Second, new food processing technology. This is because of economic reasons. One of the objectives of food processors is to produce a product in large volume and at a faster rate in a centralized plant. Handling of food in large volumes has its disadvantages. Accidental contamination of the product by a pathogen can cause a foodborne disease outbreak among large numbers of people over a large area. Third, miscellaneous factors. There is also some speculation that present methods of raising and feeding food animals and birds may have given some pathogens a better chance to become established as carriers in the animals and birds. Food from the sources has a greater chance of being contaminated with this pathogen.
I'm going to talk about the HACCP and FSO system for food safety. First, HACCP stands for Hazard Analysis Critical Control Point System. HACCP is a system that should lead to the production of microbiologically safe foods by analyzing for the hazard of raw materials, those that may appear throughout processing and those that may occur from consumer abuse. It is a proactive, systematic approach to controlling foodborne hazards. The HACCP systems place emphasis on the quality of all ingredients and all process steps on the premise that safe products will result if these are properly controlled. The system thus designed to control organisms. Next is FSO, that is Food Safety Objective. FSO is a statement of the frequency of maximum concentration of a microbiological hazard in a food considered acceptable for consumer protection. It has been endorsed by the ICMSF, and the steps that need to be taken for its development are outlined in Figure 21 until 4.